So in the process, cows have been ground up and put into the feed that has been given to other cows. So it's being passed on in that way. So we have human reservoirs, we have animal reservoirs, and of course, environmental reservoirs. So we're talking about soil, we're talking about water. Clostridium would be an example of a microbe that you would find in the environment. No way we'll ever get rid of Clostridium tetanus, Clostridium botulinum, because these things are in the soil and you just couldn't get rid of it. Legionella, another microbe that you would find in water. You can't really control microbes that are in the animal population or in the environment other than to ward off that environment, keep people out of it, eliminate the animals that cause the disease. Yeah.
So when you mentioned bats and stuff and raccoons, so there's different varieties of like raccoons, a kid with the encephalitis from raccoon feces. Bats would be rabies, right? How about like E. coli 0157 from cattle and livestock? That's zoonotic as well?
Sure. But that can also be human as well.
Right, but the cases where they're finding it coming from livestock.
In cases when it's fecal oral from animals, yes, then it would be a zoonosis.
So then the vectors of zoonotic disease are varied.
Yes, right? Absolutely. Absolutely. Now let's consider the portals of exit. So here we're talking about transmission now. The microbe has to leave the reservoir. If you think about just with regards to humans, our portals of exit, our intestinal tract is the main portal of exit for a lot of microbes. Our respiratory tract, just breathing and coughing and sneezing, another way for the microbes to leave the body. The skin serves as another means as microbes get sloughed off. And then semen and vaginal fluid can also serve as a means of transmission, a means of exit for the microbe out of the body. When we talk about transmission, it can go two ways. It can be horizontal transmission or vertical transmission. Vertical transmission is just from the mother to the fetus. We won't concern ourselves too much with that. Horizontal transmission then, transmission from one person to another person. In several ways that this transmission can take place. Contact. Now here, there's two ways that you can have contact. You can have direct contact. So this is somebody then that you are touching, shaking hands with, intercourse. Yes, that's contact. Seriously. And then indirect contact would be hard surfaces, fomites. These are things then that the microbe gets transferred to. So doorknobs and things like that. Bathroom doorknobs serve as a great fomite. Indirect contact. A lot of this means of transmission we see with fecal oral microbes require this kind of contact. We also have droplet transmission, which is considered a kind of direct contact because it requires me to spit on you. No, seriously. I have to be, or you have to be within close contact that your sputum that can be inhaled. Usually about within 3 feet. Okay. 3 feet's enough to do droplet transmission here. So these are ways that we can transmit using humans, if you will. But we can also transmit via water, food, air. So transmission via food and water. We know animal products, mad cow disease here, animal products harbor microbes. So they can serve as a source of contamination, a source of infection, if you will. And in fact, cross-contamination is the main means by which we infect ourselves with our food. You can have outbreaks that occur through waterborne microbes. Vibrio cholera is an example of that you can find in your water. Generally, our treatment facilities, chlorination and stuff like that, do a pretty good job of killing most of the microbes that you'll find in our water. That's not to say that your tap water is microbe-free. There are microbes in there, but any of those that would cause disease have been eliminated through treatment. Air can serve as a means of transmission. Now we're talking about microbes that are going to enter the lungs. And they can do so by a process called droplet nuclei. Not only do I spit on you when I talk to you, but some of that stays suspended in the air. And it'll dry a little bit, and the bacteria that are adhered to those little tiny droplets, as it dries, they kind of float freely and get moved around with the air currents. More people in a room, the more bacteria you got circulating in the air. So we've got a lot of bacteria going on right now in the air here. So those are suspended up there in the air. Doesn't take much then for you to inhale it. And then you've been infected with the microbe. Yes?
Is droplet nuclei that process, or is it what you would name those microbes?
I would say that droplet nuclei is the transmission process. And you would, yes, you would name those little things droplet nuclei, but you would also use the term to apply to the process. And then vectors are another means of transmission. Now we're talking about living organisms that are carrying the disease. And we talked about early in the semester 2 types of vectors. You can have mechanical vectors and you can have biological vectors. Now a mechanical vector is one that will pick up the microbe and bring it to you, or bring it to your food. And we talked about flies serve as great mechanical vectors because they land on things, poop and other stuff like that, and then they carry it into your house and land in your sugar bowl or some other place. And so they are great mechanical vectors for carrying microbes. A biological vector then is one that has to bite you in order for the transmission to take place. Fleas and ticks are examples of biological vectors. With this here, if you can control the vector, you can control the spread of the disease. One of the things that we're trying to do in Africa right now is control the vector, control the mosquito, and you can control the spread of malaria. Portal of entry. All I'm going to say about the portal of entry is that it needs to be appropriate. Okay, so if we're talking about a microbe that is spread via fecal oral, then you have to drink it or you have to ingest it. Inhaling it is not going to make you sick. So, for instance, in lab with E. coli, you know, if you had a culture of E. coli and you sat there and you inhaled all day long, sniffed up, probably not going to make you sick. Or some other microbe that is spread via fecal oral. But if you drink that culture, now we got an issue. Okay? Same with the respiratory system. Now, if we're talking about a microbe like tuberculosis that causes a respiratory infection, you know, drinking tuberculosis isn't going to make you sick because it doesn't infect you that way. The portal of entry has to be appropriate for the infection. What are some factors then that influence disease? The dose is a main contributor to the spread of disease. In most cases, if you are exposed to small doses of microbes, your immune system is to respond accordingly, and you're going to be able to defend those small exposures. But large exposures generally overrun the immune system initially. And so they take hold, and that's why you get sick. And that's why the infection takes hold. And it's not until your immune system has a chance to recover that you get well. However, that's not to say that some microbes don't cause infection with low doses. As we will see, Staphylococcus and Streptococcus, both of them are strains that can cause infection with just as few as 30 cells, depending on conditions. Vibrio cholera, on the other hand, needs, hundreds of thousands of cells in order to cause an infection. There's an incubation period that will influence the spread of a disease. So, if the microbe expresses itself, the disease manifests itself very early on, within hours of being exposed, then we know right away who's sick, or very early in their exposure who's sick, and we can quarantine them and slow the spread of the disease. But if the incubation period is longer, then we don't know that that individual has been exposed, is infected, and possibly spreading the disease. So the longer the incubation period, the greater the chance that the disease can spread to other individuals. Other factors that influence population characteristics. What is the immunity of the population? If they've been exposed to this microbe before, then maybe it doesn't affect that many individuals in the community. But if it's a new microbe moving through the community, then many of them could become sick. What's the general health of the population? Are they malnourished? Are they fatigued? What about the age? The elderly and the young are going to be more susceptible. The young because they don't have their immune system built up yet, and the elderly because their immune system is becoming depressed. Gender, religion, genetic background, all of these can play a role in whether or not a disease has the ability to spread.
Can you say what's the immunity of the population? You also mean like certain immunity too?
I'm sorry?
When you say what's the immunity of the population, you're including herd immunity into that too?
Current?
Herd immunity? Let's talk about herd immunity.
Going back to
Herd immunity, yeah, absolutely. Yes, yes, absolutely, herd immunity. I wasn't hearing the herd part, like. So yeah, if you've got a community that has been vaccinated like that, and 95% of them are, then you will have a difficult time, a disease would have a difficult time spreading through that community. But if you find one that's only 50% vaccinated, then it's going to spread through much easier. So epidemiologists, they have to investigate disease outbreak, and there's 3 different studies that we can talk about. I'm just going to mention them briefly here. So the investigation of an outbreak starts with what we call a descriptive study. And a descriptive study basically is just gathering facts about the infection, characteristics of the people that are involved in the infection,